Okay, I'd like to talk some more about the retina and now talking more about rod and cone cells specifically. So one of the classic questions, or one of the things you might be asked to explain in this process, is the process of signal transduction, or how transduction takes place in rod and cone cells. And it's very similar how it takes place in both types of cells. So I'm only going to explain one, and in most cases you might only be asked to explain one or the other. So my question is, explain how transduction takes place in rod and cone cells in the retina. So the first thing that happens is a photon of light hits retinol, or retinal rather, which is embedded in the opsin protein. Okay, so there's this retinal, it's embedded in the opsin protein, and it's going to absorb a photon of light. It's going to absorb some of the energy. So the retinal absorbs some energy from the photon of light, which causes the retinal to change conformation. Okay, this is a classic situation in biology where you see some form of energy causing some conformational change in a protein. And that's essentially what's happening right here. So there's going to be a conformational change of retinal, and the conformation is going to change. Okay, it exists normally in a cis conformation, and it's going to change to a trans conformation. Okay, so it's going to go from cis to trans. And what that conformational change is going to do is it's actually going to cause a conformational change in opsin. Okay, so the conformational change in the retinal will cause a conformational change in opsin, which is the protein it's embedded in. And this is going to move on to initiate basically a biochemical cascade. Okay, and a biochemical cascade is like any other cascade. It's like maybe you might recall a phosphorylation cascade or something like that from biochemistry. And it's very similar. It's basically a series of events that occurs that leads to some form of change. Okay, something's going to happen. Something's going to be, some gene is going to be initiated, or a transcription of some gene is going to be initiated, or something's going to happen. Okay, but the bottom line here is that the conformational change leads to a biochemical cascade. Okay, and in this case, the result of the cascade is the production of something known as cyclic GMP. Okay, phosphodiesterase. Okay, cyclic GMP phosphodiesterase is the result of the biochemical cascade. And the cyclic GMP phosphodiesterase breaks down cyclic GMP. And you might say, well, what's the point of breaking down cyclic GMP? Well, this results in less cyclic GMP being available, and thus less cyclic GMP-gated sodium ion channels being open, okay? And the reason why that's important is it results in a decrease in membrane potential, okay? It results in a hyperpolarization. And thus, this leads to a decrease in the release of the neurotransmitter from the rod cell.